The new Omicron COVID-19 variant is more transmissible than the Delta variant, but a booster vaccine dose seems to be effective against it, according to a new study by an international team of researchers. The study, supported by the European Union's Health Emergency Preparedness and Response Authority, HERA, showed that the blood collected from patients infected with COVID-19 12 months ago and those vaccinated against the virus 5 months ago barely neutralized the Omicron variant. But the sera of individuals who had received a booster dose of Pfizer, analyzed 1 month after vaccination, remained effective against Omicron. To reach this conclusion, scientists from the Institut Pasteur and the Vaccine Research Institute, in collaboration with KU Leuven, Belgium, Orleans Regional Hospital, Hospital European Georges Pompidou, APHP, and INSERM, studied the sensitivity of Omicron to antibodies compared with the currently dominant Delta variant. The scientists from KU Leuven isolated the Omicron variant of SARS-CoV-2 from a nasal sample of a 32-year-old woman who developed moderate COVID-19 a few days after returning from Egypt. The isolated virus was immediately sent to scientists at the Institut Pasteur, where therapeutic monoclonal antibodies and serum samples from people who had been vaccinated or previously exposed to SARS-CoV-2 were used to study the sensitivity of the Omicron variant. The scientists used rapid neutralization assays, developed by the Institut Pasteur's Virus and Immunity Unit, on the isolated sample of the Omicron virus. This collaborative multidisciplinary effort also involved the Institut Pasteur's virologists and specialists in the analysis of viral evolution and protein structure, together with teams from Orleans Regional Hospital and Hospital European Georges Pompidou in Paris. The scientists began by testing 9 monoclonal antibodies used in clinical practice or currently in preclinical development. 6 antibodies lost all antiviral activity, and the other 3 were 3 to 80 times less effective against Omicron than against Delta. The antibodies bamlanivimab/etesevimab, a combination developed by Lilly, casirivimab/imdevimab, a combination developed by Roche and known as Ronapreve, and ritonavimab, developed by Keltrion, no longer had any antiviral effect against Omicron. The tixagevimab/silgavimab combination, developed by AstraZeneca under the name Evusheld, was 80 times less effective against Omicron than against Delta. We demonstrated that this highly transmissible variant has acquired significant resistance to antibodies. Most of the therapeutic monoclonal antibodies currently available against SARS-CoV-2 are inactive, commented Olivier Schwartz, co-last author of the study and head of the Virus and Immunity Unit at the Institut Pasteur. 5 to 31 times more antibodies were nevertheless required to neutralize Omicron, compared with Delta, in cell culture assays. These results help shed light on the continued efficacy of vaccines in protecting against severe forms of the disease. We now need to study the length of protection of the booster dose. The vaccines probably become less effective in offering protection against contracting the virus, but they should continue to protect against severe forms, explained Olivier Schwartz. The scientists concluded that the many mutations in the spike protein of the Omicron variant enabled it to largely evade the immune response. Ongoing research is being conducted to determine why this variant is more transmissible from one individual to the next and to analyze the long-term effectiveness of a booster dose.